Hello friends, welcome to BMH Learning. This video is about lactate dehydrogenase assay. When the cell membranes are compromised, or you can say damaged, lactate dehydrogenase is released into the surrounding extracellular space. LDH is soluble but stable enzyme present inside every living cell. Now, only way for the LDH to come out of the cell is when the cell membrane integrity is compromised. So this means the presence of this enzyme in the culture medium can be used as a cell death marker. So lactate dehydrogenase assay is used to measure either the number of cells via total cytoplasmic lactate dehydrogenase or membrane integrity as a function of the amount of cytoplasmic LDH released into the medium. So this way, the relative amounts of the live and dead cells within the medium can be quantitated by measuring the amount of released LDH. Now, to measure the amount of released LDH, both colorimetric or fluorometric cytotoxicity assay can be used. In this video, we will talk about colorimetric assay. LDH cytotoxic colorimetric assay principle. While using an LDH colorimetric assay, the amount of LDH released in the medium is measured with an enzymatic reaction which involves the conversion of idonitrotetrazolium into a red color formazan. The compound is actually a tetrazolium salt. The reaction is carried out in two steps. In the first step, when LDH is present in the cell culture, it reduces NAD positive to NADH and H positive through the oxidation of lactate to pyruvate. Basically, the enzyme is the oxidoreductase and catalyzes the interconversion of lactate and pyruvate. In the second step, diaphorase, which is used as catalyst, then transfers H positive to the tetrazolium salt to form the red colored formazan salt. The amount of color produced is measured at 490 nanometer by standard spectroscopy. Also, the amount of color produced is proportional to the amount of damaged cells in the culture. So that means more is the cell damage, more intense would be the color. Due to the inherent linearity of the LDH cytotoxicity assay, it can be easily used to accurately determine the percentage of damaged, or we can say injured cells in a sample. Moreover, its simplicity, reliability, and accuracy makes it an excellent alternative to chromium release cytotoxicity assays, which is a highly sensitive method that measures low levels of cytotoxicity by using radioactive isotopes. The assay can also be used with different cell types to determine cell-mediated and chemical-mediated cytotoxicity. Apart from this, the assay is ideal for cell-free supernatant from cells in culture. Materials required: reagents, acetic acid, hydrochloric acid, idonitrotetrazolium chloride, lithium L-lactate, MPMS, that is methoxyphenazine methosulfate, NAD, tris-base, splice, 96 well plates, centrifuge tubes, microcentrifuge tubes, reagent reservoirs. Equipments needed: single channel and multi-channel pipettes, analytical balance, magnetic stir plate, pH meter, orbital shaker, plate reader for measuring absorbance at 490 nanometer wavelength. Now, regarding the procedure, the protocol for the assay would depend on the type of cell lines you are working with or other experimental needs and conditions. So you can follow a certain standard protocol which you can find in research articles or some other books specifically made for biochemical tests. Or an easy alternative is the commercially available kits for the LDH assay, and you can follow and perform the protocol assays according to the manufacturer's instructions. This was all. Thanks for watching.